And our next speaker is Jeff Wintersinger. That was you, right?
Yeah. Thank you. So hi, I'm Jeff. I'm part of Quaid Morris's group in Toronto, and I'm going to be talking to you today about Pairtree, which is a new algorithm we've developed to do fast phylogeny reconstruction using multiple cancer samples. So I won't dwell on the background terribly much. Suffice it to say that cancer within a patient is not a homogeneous disease. We have multiple genetically distinct subpopulations. Having established that, we can start to think about taking tissue samples from the patient. So suppose early, perhaps right around the time of diagnosis, we obtain a tissue sample from the patient that would be a mixture of these two subclonal populations, this founding clonal teal population and a subpopulation in green that descended from it. Then perhaps we treat the patient gets better, but patient relapses. At time of relapse, we get another tissue sample with a different mixture of subpopulations. And we can even think about taking a third tissue sample, perhaps from a different spatial site in the patient. So if there was a metastasis, we might also get a sample from this. So we end up with 3 different tissue samples, each of which is composed of a mixture of different subpopulations. Now, the data we're interested in are mutation read counts. So for each of these samples, we can do genomic sequencing, we can do variant calling. In this instance, we are saying that the reference position A has changed to a G, and this has happened for 11 of the 24 reads that we have. So we can get the frequency of that mutation. But the important thing is that when we do this sequencing, we're getting a convolved signal from all of these combined subpopulations. So we have to think about deconvolving. So our goal ultimately is to take these read counts from each of our samples and then build a tree. You'll note that this tree maps exactly to the picture I showed you before. So we have our founding teal cancer cell population that gives rise to these others in a linear pattern, and then we have 2 distinct branches of evolution being explored in parallel. Now, there's a couple of points to make. The first is that every node in the tree represents a genetically distinct subpopulation. So we assume that you always inherit the mutations of your ancestors, but each of these nodes representing a population also has its own set of mutations that distinguish it from all the other populations. Second point is that, of course, each sample consists of different proportions of each of these populations, but the really important thing to understand is that there's only a single tree that describes the complete evolutionary history of disease in the patient. So this means that some of our populations on the tree will have 0 frequency in some of our samples, and that's fine. For instance, our red metastatic population here is going to have 0 frequency in sample 1 and sample 2 because it just didn't exist back then, but that's fine. There's one tree that describes the full disease. So the story I'm going to tell you is about data that our collaborators in Toronto came to us with. So they had 14 acute lymphoblastic leukemia patients, and we spent a lot of time trying to build trees for them. They had, for each of their 14 patients, tissue samples from diagnosis and from relapse. But the interesting thing about their project is that they also had dozens of patient-derived xenografts for each of these patient samples. So they'd take each patient tissue sample, and they'd have dozens of mice into which they'd implant the cells. And then this meant that in the extreme, for one patient, we could have 90 different tissue samples representing the cancer at different stages of evolution. So I don't have time to delve into the tree, but suffice it to say, this was exome sequencing, so we only had about 100 mutations for this patient, but we were able to recover 17 distinct subclones and build this really high confidence tree. And you can start to read an interesting story from the tree because we saw these 2 founding clonal populations that were present at diagnosis and relapse, but we had a diagnosis-specific branch that had disappeared by the time of relapse. Then we had this relapse-specific branch that was present at very low frequency for just the head of that subtree. at the time of diagnosis. But by the time of relapse, perhaps in response to treatment, it had come to dominate the cancer and give rise to populations 7 through 9. And these in turn gave rise to 4 distinct branches at low frequency that were starting to come up. And if the cancer continued to evolve, presumably one of these would come to dominate. Because remember, this is a liquid cancer, so it's not as subject to neutrality as you might see in solid tumor. So what we did is we tried a number of algorithms that were already available, and all of them failed for various reasons. So we sat down and thought about how to exploit the richness of this data, and we came up with this algorithm, Pairtree. Now, the key contribution we make is to recognize that if you want to build a tree, all you need are pairwise relations between mutations. So let me explain. If you come to me with mutations A and B, there's only 4 possibilities. So the first is that they occur in exactly the same set of cells. We say they're co-clustered. The second would have A being ancestral to B, so that all cells that have B must also have A, since they inherit their ancestral mutations, but we'll have some cells that have A but not B. Then there's the inverse as a possibility, where B is ancestral to A, and then there's the possibility that A and B are on different branches, so that no cells have both A and B together. Now, we are making the infinite sites assumption, so that is, we're saying that single genomic sites will never be mutated multiple times. However, we can detect violations of this assumption. If you're interested in that, talk to me later. So the claim I'm going to make is that all you need are pairwise relations to get a tree. So if you come to me with this matrix, and that tells me which of those 4 relations every pair of your 4 mutations, A, B, C, D, falls into, I can give you a tree. Likewise, if you give me a tree, I can give you that matrix. So this is an if and if, or rather an if and only if kind of relationship. Now, the second half of this is trivial. If you look at the tree, you can read off, oh, well, A is ancestral to D, B and C are different branches, and so on. To get the first half, we need to infer from data. So the way we do that is we say, if you have mutations A and B, and you have 3 tissue samples, S1 through S3, if we see that the frequency of A is always higher than B, then we can be pretty confident that the correct relation has A being ancestral to B. Conversely, if you came with 2 other mutations, C and D, and we saw C was higher than D in sample 1, but D is higher than C in sample 2, we can say neither can be the ancestor of the other, so different branches is the right way to look at this. Now, this is probabilistic. We get a probability distribution over these pairwise relations for every pair, because we do have ambiguity sometimes in the correct relation. This creates ambiguity in the resulting trees. Now, if we go back to those 14 ALL patients I was telling you about, we can characterize the quality of solutions that we come up with relative to other algorithms by asking, relative to some truth, some true tree, how well do the pairwise relations matched by our trees or other methods' trees match the pairwise relations implied by that ground truth? So what we did to get that truth is we built a really high-quality tree over the course of a bunch of weeks of effort that was extensively refined by hand. And we're asking, how well can we recover that? Effectively, what we're saying is that we have almost 0 error in Pairtree for 11 of those 14 patients. Other algorithms don't do as well. Now, of course, this is kind of a biased evaluation because we're evaluating relative to this baseline that we have provided. So we can also evaluate on simulated data. So we did several hundred simulations where we're generating with lots of different characteristics of trees and asking how well, again, can we recover those pairwise relations via our trees relative to the ground truth tree. What we see is that Pairtree does really well, especially in the case where you have many tissue samples, where you have 30 or 100 tissue samples providing different estimates of these frequencies of mutations. Other algorithms don't do as well. And so what I'm not showing you here is that actually a lot of the other algorithms we try, when we increase the complexity of the problem we're asking them to solve by giving them more subclones or more tissue samples, many algorithms in that setting fail to give us any answer at all. And so what we think is happening is that the more tissue samples you have, the more constraints are placed on your tree. And this makes the search space over trees very difficult to navigate. So what we do to overcome that is we explicitly model these pairwise relations, and we use that as a stepping stone. So we go from data to pairwise relations and from there to trees. That helps us considerably. So with that, I would like to thank my supervisor, Quaid, and our collaborators, Stephanie and John. If you'd like to chat more, I'll be at poster K49 tonight. So thank you for your time.
Do we have any questions for Jeff? Ed over here.
So it strikes me that you have an additional constraint in your data when you have these multiple samples, which is for secondary tumors, you have a spatially segregated system. And do you think that could provide additional information in this context? And are you using that information?
Right. So in that instance, we might observe in a metastasis that was seeded by one subclone. We wouldn't expect to find any presence of those other subclones. And so we don't explicitly model that. Hopefully our data is good enough that it's telling us that we don't actually have those sorts of mutations, but we could certainly provide that as an additional constraint that would constrain the space of trees that we're looking at.
One more question over here, and then we continue.
Yes, so part of the reason we think we see such sharp selection in the example I showed you is because the patient was treated. And for what it's worth, the xenografts were also treated, so we'll see some extent of that in the xenos as well.
Okay, let's thank the speaker again. And with this, I leave the microphone to the next chair, Q.
Hi, good morning. Now, the next speaker is your work. So he's using the local coevolution information to identify one type of very important